Hi everyone, my name is Rose Leeger and my project is titled Using Antarctic Surface Currents to Understand Antarctic Toothfish Life History Traits. So first, a little bit about the location where my PhD is studying is Antarctica, which as we know is at the bottom of the world, which allows for some really unique geography and climate for some truly amazing organisms that reside there. The two seas that I look at are the Ross Sea, which is closest to McMurdo, that's the United States' largest field station. And some unique oceanography features that occur there include a deep continental shelf, which allows for some very deep water, and it's really important for the circulation of nutrients. It also has a wind-driven gyre that rotates clockwise and is formed by the interaction of the ACC, or Antarctic Circumpolar Current, and the continental shelf. A really unique characteristic about this sea is the formation of the MPA, or Marine Protected Area. This marine protected area protects over 1.5 million square kilometers and is the largest MPA in the entire world. This is formed mainly to being due to the most pristine ecosystem. It's furthest away from pollution, mining, invasive species, and some fishing, and it has really rich biodiversity here. The next sea that we'll look at is the Weddell Sea. This Weddell Sea is up near the Antarctic Peninsula, close to South America. And if you've ever heard of Shackleton's boat, the Endurance, this is where it got trapped and crushed by ice. Here it also has a gyre that flows from a northward current, and it plays a huge role in ocean water modification and deep water formation. In this gyre, we do have weak surface currents, so deep circulation is more important in the dynamics of deep water movement. And again, there's lots of biodiversity, including the largest colony of Adelie penguins, yet there is no MPA in place here. Now, some of the biodiversity I just mentioned are listed below. So first, we have the silverfish. It's a keystone species, and it's pretty small, and it's the most abundant pelagic fish. We also have the longfin ice devil, the only member of its genus, and there's not really too much more information about them other than that they've been found or discovered. And lastly, we have the Antarctic toothfish. And this is our top fish predator, and it's huge, up to 6 feet long, and they feed on almost anything. They're very long living, they have a slow reproduction. And the reason why all of these fish are so special is that none of them have a swim bladder. Instead of a swim bladder, they have antifreeze glycoproteins, which help them thrive in these sub-zero Antarctic waters. Lacking a swim bladder means that these fish are neutrally buoyant. Some of the larval and pre-reproduction stages may be negatively buoyant for better protection. So a lot of studies have shown that the use of gyres for egg transport and spawning grounds could be important. The storage of all these lipids is super important. You'll see why later. And my PhD looks at the toothfish and how we can just better protect them. So unfortunately with the toothfish, something that's come about in the early 1990s is fishing. The reason why they're fished is because of those lipids and the presence of the glycoproteins is that they're very tasty. It's pretty hard to over undercook these fish. And they're also resold as Chilean sea bass, even though they're not Chilean or bass. And this can be found even in our local markets like Whole Foods just right down the street. So there's been a lot of countries that are fishing this species, and there's been a lot of pirating and just illegal issues going on, and also the fact that now they're being taken out of Antarctica at a really rapid rate. And the biggest issue with all of this is, of course, overfishing. How much are we really changing this toothfish population with the fishing industry, and how can the toothfish life history be changed, especially as one of the longest living fish in Antarctica? Here's an example of probably one of the only videos that exists of a toothfish, since I do live down deep. But we can see that toothfish do use neutral buoyancy. A unique characteristic about this that we know from oceanography is that because of the lack of the swim bladder, they rely completely on the density of the water column to forage, reproduce, and other important life history characteristics. Now, as we know, deep ocean currents are driven by differences in a water's density, which we know is controlled by temperature and salinity. The thermohaline, which is a global circulation driven by convection at polar latitudes, is really important for these fish, and it allows for movement seasonality, and it could induce populations to change or even migrate over time. Now, while the adult toothfish do use the thermohaline in order to move around the water column, the interesting thing is their eggs don't. Unfortunately, we don't have a picture of toothfish eggs, so here's a rendition of it. But these toothfish eggs that they lay are positively buoyant and they're pelagic. So they float on the upper layer of the ocean, and they can float for up to 9 months. And they're just completely driven by the surface currents for transport. So what I want to look at is to see can we track toothfish egg pathways to better understand areas where they could be possibly protected from overfishing. Now there's a couple things to note. So we want to still understand sea surface currents, since that's what's going to be driving the gyres and possibly moving these eggs all around the Southern Ocean. However, we also want to look at thermohaline seasonality. We want to make sure that the adult fish that are laying these eggs are able to actually go throughout the water column, that there's not any outliers, anything going on that could happen year to year. And also we want to consider the areas of fishing past and present. Where are these fish actually being fished, and how can we understand where the eggs could then end up, could relate to their survival? So lucky for us, we have a perfect data set that can do this, and that's the ECHO data set, or the Estimating the Circulation and Climate of the Ocean. They look at 3D and 2D varying ocean and sea ice states, and their claim is that they're a little different from other ocean models by reproducing observations in a physically and statistically consistent manner. They're really important because over 1,000 papers and publications have looked at things like sea level rise, sea ice loss, EMSO events, the water and carbon cycle. So with Chris's help, we thought this was a really good indicator of what we're looking for. And it should be noted, just like what we learned in class, that we're going to kind of look at the differences of density and salinity for the thermohaline. So first, just the Southern Ocean currents as a whole. This was just kind of mapping and understanding where they looked at. So the ECHO monthly surface currents go from 1992 to 2017, and that timeline works really well for us since the beginning of the toothfish industry began in 1997. So we can kind of use this data set to look at the beginning of the fishing industry as well as the continuation. We currently have samples from all the way from the 2022 fishing season. So next, we wanted to look at the different aspects of the thermohaline circulation. So as you can see here, as salinity is decreasing, the average air-sea latent heat flux increases. With saltier water, it becomes denser, so it sinks. And we can see here in this map that it kind of sinks between January and February, which is a common time that the fisheries are catching these toothfish. We also see that there's really no differences seasonality from like the early fishing industry all the way to 2016. It seems like the temperature and salinity follow each other with no major outliers. Once again, we're now going to look at the density stratification, and this is looking at the wind stress stratification, the meridional wind stress east to west, and also looks at the overall density stratification. So now this is kind of the model combining the temperature and salinity to understand that. We do know that if density goes too deep, toothfish could be at a much deeper depth, which is less likelihood for those eggs and larval fish to actually reach the surface and survive. So we can see here that it does fluctuate year to year, but unfortunately our fishing boats have gotten really good about catching toothfish at really deep depths, and that seems not to be too much of an issue. So next we wanted to look at Weddell Sea surface currents. So this is, again, with Chris's help using MATLAB, kind of just an overall understanding of where the Weddell gyre is and how fish would use it. So we kind of imagine, I know it's an adult fish here, but imagine that these are its larval eggs using the sea surface currents to kind of use the Weddell gyre and just kind of circulate around. We can see this here using laryngetical particle tracking. Again, thanks Chris for this, because I had no idea what that was. And just looking at the sea surface currents in the ECHO data set timeline and adding in different latitudes and longitudes to see the predicted movement. So for this data, we looked at it daily. Monthly apparently would be a little too jumpy, and the tracking begins by looking at a possible Latin long that it could be, and then it just goes through that time series. So for the most part, we used mainly 1 to 2 years. I wanted to really see how their original location could influence where they ended up. Eventually, as they grow in the water column, and then how they could eventually be hunted by the fishing industry. So this is kind of like a model and a perfect example of a particle, which is that egg, using the Weddell gyre for this tracking. I tested different larval spawning grounds that they could also use. So one of them is, again, that particle in the center of the gyre, kind of really using that as its movement. Another one was a particle a little too close to the shelf. Should be noted here that we did have some code where the experiment would be terminated if the particle reached land. But we can see here that it's less likely to go to the open ocean. And then lastly, a particle that's a little bit more west, so we kind of move the map a little bit more. And we can see here that the particle actually ends up a little bit further away from the gyre and possibly into that area where fishing occurs. Now, the reason why we're looking at this is currently where fishing is. So as we can see, the closer the particle is to the continental shelf, or at least within that main gyre system, the more likely it is to possibly be away from some of that fishing activity. We then did the same for the Ross Sea surface currents. This one's a little bit trickier because our map kind of cut the Ross Sea in half, but we can see the main part of the gyre below, with once again our toothfish eggs using a similar end part. Here we looked at two different scenarios. So one of them was the particle once again centered in the middle of the gyre, mainly seeing that it does use that current. And the other one was the particle on the outer edge. And we see that it flows way beyond the shelf. And the answer to where fishing occurs within the Ross Sea is a lot more places. So if the Ross Sea has the most amount of fishing present, part of it was that the Weddell Sea has a lot harsher conditions, so they're less likely to find these toothfish. The Ross Sea is more protected. But do not worry, because the MPA is where this is located. So the MPA is divided into 3 different zones. There's a general protection zone, which is closest to the continent, a special research zone in kind of the middle, and a krill research zone in the outer, like western edge. And a lot of guidelines have been in place, with the most strict being the general protection zone, which is that larval closest to the continental shelf. So as we can see, possibly with the larval being found closer to the shelf could mean better protection. So seeing how these eggs are using the currents can better help us understand how to better protect the species. So there it is. So overall, some conclusions is that different areas of the Weddell and Ross gyre can indicate species success. We know that in both gyres, having the particles or eggs being centered or near the shelf could lead eggs further from fishing activity. We also know that larval egg success can be further translated into seasonal fluctuations of the thermohaline circulation, allowing for those adult toothfish to possibly continue and allow them to give their eggs possibly further distance away from the fishing industry. And obviously, understanding life history of any organism, especially those deep-dwelling, long-lived fish, is really hard. And a lot of gaps remain that we don't really know the fact about how often these toothfish are spawning. Some studies say that doesn't even occur yearly, so that could really change how we look at things. A lot can happen in between the eggs to the gyres and maturity, and maybe does the size or the scale of the eggs matter? Could they be using other transport systems we aren't sure of? There's a lot to discover. So yeah, overall, thank you for coming. You can feel free to reach out and ask any questions. Here's my email. And luckily, we're doing a lot more chemistry with this to kind of answer some of those questions. So looking at ocean models is really exciting because that's something that wasn't really in our original plan, but we're hoping to kind of combine this knowledge we've learned in class to figure out truly what these fish are doing. And here's my references. Thanks.